Okay, so we are on. Welcome to everybody with this new edition of the Legumes Society webinars. Today we are here with Paolo Annicchiarico, that most of you probably know. He's the previous president of the International Legumes Society, also a colleague of mine. And today we are going to talk about intercropping and its value for forage and grain legumes. Please, Paolo, start with your presentation.
Thank you, Nelson, and good afternoon, or perhaps good morning to everybody who is listening and taking part in this webinar. What is intercropping? It is defined as the simultaneous growing of two or more crops in proximity on the same land across the whole cropping season or a substantial part of it. The first scientific reference to the value of intercropping was provided by Charles Darwin in The Origin of Species. He was actually mentioning an experiment that took place over 200 years ago in a botanical garden in the United Kingdom. I'm showing you here the plan of this large experiment where there were many different mixtures, and the result that Darwin cited was that when we mix grass species, we get on average a higher yield than growing them individually. But actually intercropping was known since many centuries by different peoples. For example, in Central America, Maya were growing using their typical milpa, that is a three-crop system, intercropping maize together with common bean and squash. I guess we all know that intercropping increases the crop yields, for example, as measured by the land equivalent ratio, and the yield stability by better utilization in time or space of resources like water and nutrients. This is by a complementary foraging pattern of the associated species, by reduction of pests and diseases, mainly by a dilution effect of the host density, better control of weeds, and then ability of some species to produce or mobilize and make available for the companion species one or more limiting soil nutrients, such as nitrogen in the case of legumes, but also phosphate, for example, by lupins, etc. And in addition, intercropping provides important agro-ecosystem functions, especially when it is based on legume crops, such as nitrogen biological fixation, improvement of physical, chemical, biological soil fertility, reduction of environmental friendly inputs. Now, why intercropping, which is a pillar of agroecology, is after all so little used? There are a few important challenges. One is the availability of suitable machinery for planting and neat separation of the harvested seed. This is particularly for grain crops, but we are progressing on that. And then there is the difficulty of cropping reliably balanced mixtures. And this should be achieved through breeding and optimal crop management. So that today we are focusing in particular on the selection of material, of legume material for intercropping. You see here, for example, different solutions, and there might be many others. For Lucerne in France, this is our own work on white clover, red clover selection in Ireland. There is also an issue of farmer evaluation, participation. It is difficult to breed for intercropping. First of all, because we have a potentially huge number of possible companion varieties for a new variety. And then different crop managements may affect competition dynamics. such as the nitrogen amount and timing, and the sowing rate of each component, the sowing pattern, for perennials, even the crop exploitation. So that the key questions for the breeders are, I believe, first, is there a substantial consistency for genotype performance across pure stand and mixed stand? Because if there is, we can just select under pure stand. In case we need a specific selection for intercropping, could it be based on useful adaptive traits that we could observe in pure stand? Because cropping in pure stand is normally less expensive than growing evaluating in mixed stand, where we need botanical separation. Third, does the genotype suitability to intercropping vary largely depending on the associated species and variety? And this is a further complication. And finally, is the genotype suitability to intercropping strongly affected by crop management, meaning that we should select across different managements, or we should choose carefully a target management? So I'm going to talk about all of these questions today. This guy used considerable time and resources to verify whether the Earth is flat or round. My suggestion is not to do like him, and to profit instead of all the available past experimental and theoretical work, which is plenty. For example, nearly 80 years ago, there was this study focusing on the selection of white clover for intercropping. And then in the late 60s and the early 70s, there were several really inspiring papers. I'm reporting here a few that I found most valuable. And now the problem in understanding how to breed for intercropping is that in the end, there aren't many really useful experiments where we are evaluating a pretty large set of genotypes of one species in pure stand and in mixed stand with a good number of genotypes of the other species. Because these kind of experiments, they are quite demanding. They require a lot of plots. Nearly 30 years ago, I performed one such experiment, and I would like to use it as a cornerstone for my presentation, because there I found many results that were then verified in several other earlier or later smaller experiments. In this experiment, there were 6 white clover genotypes associated with 8 grass varieties from 4 different species. They were grown in pure stand, mixed stand, under moderate nitrogen fertilization. And I'm talking about yield of clover and grass over 2 years. First of all, it's useful to see the mean values of the clover genotypes and the grass varieties in terms of clover yield and grass yield. And first of all, we can see that the clover is generally at competitive disadvantage with the grass. You see here, on average, the yields of the clover compared to the yield of the grasses. And this happens pretty frequently in legumes. The second thing that we could see is that increasing on average the clover yield does not imply a decrease of grass yield. You see, it remains pretty constant. because normally the clover does not really affect so much the competition with grasses. It is not competing for a key resource such as nitrogen, and actually in forages is even providing some nitrogen to the grass, meaning that greater legume yield also means greater total yield of the mixture, which is not the same for grass varieties. You see, the grasses that have a higher grass yield, they also have a lower clover yield. You could draw a line here. And now what happens to the responses of the individual genotypes in the different associations? It is a matter of genotype environment interaction. And indeed, I analyzed the data according to joint regression modeling, which is a popular model for analyzing G-by interaction. So you see here the yield responses of the 6 clover genotypes as a function of the mean clover yield in the different environments, each one represented by a condition of growing. Here we have pure stand of white clover, so the highest yield on average. And then we have association with grasses that are, here they are not so competitive, and here they get highly competitive, so that the clover yield on average is very low. And we can see that the responses of each genotype in each point here depends on the yield potential of the clover and then its competitive ability. You may think like tolerance to competitive stress. When we have pure stand, no competition, all depends on the yield potential. But here, with increasing competitive stress, then the tolerance to competitive stress gets important. And we do find genotype environment interaction of crossover type with rank changes among the top yielding genotypes, so that the genotype environment interaction effects are wider across conditions that are contrasting for extent of grass competition, nil here as very high here, or if you like, for extent of the biotic environment that it is occupied by the grass, nil here and very high here. Interestingly, the genotype environment interaction is not affected by the grass species, because, for example, we have a variety of cocksfoot here, very aggressive, and another little aggressive here. So it's just a matter of how competitive is the grass. And what about the grass genotypes? The grass is the dominating partner. And here you see that passing from pure stand to association with different clovers, the ranks of the genotypes for grass yield, they remains substantially consistent. Only when we have a more competitive white clover, we have an increase, let's say, of the difference between genotypes. It is a quantitative genotype environment interaction, meaning that in this case, we could select for intercropping just considering the response of the material in pure stand. While when we get to white clover, it is not the same. It really depends on the level of competitive stress that we are meaning. And since really the yield of the intercrop depends here mainly on the response of the non-legume species, so we want to use a vigorous grass companion, then we have to target to have high yielding mixtures, a condition with high competition from grasses, meaning that here we cannot reliably select, so for this condition, considering the responses in pure stand. Now I'm adding up suggesting evidence from other work that it is consistent with the cornerstone experiment of mine that I presented. For example, Harper, many years ago, he observed that, considering many different experiments, that the yield efficiency of a mixture depends mainly on the performance of its weaker partner. Just like we saw for the large reference experiment, no? Where having a competitive white clover meant higher mixture yield. And I'm adding here some results for pea cereal intercrops producing grains, which are just showing the same. So also for pea, normally it is at competitive disadvantage with cereals. You can see here for two data sets, independent genotypes and years, you can see the range of P proportion of these numbers of genotypes. Here is very large because we are just taking random lines from reels. Here are elite lines, but still, you see, we have a pretty large range of P proportion. If you like, a pretty large variation for competitive ability, and having greater P proportion in a mixture also means greater yield of the total mixture, that is legume plus cereal, in both cases. Then, Connell, considering many experiments, including material in the wild, concluded that asymmetrical competition is more common than symmetrical competition, where symmetrical means a balanced mixture in intercrops. So what we saw for white clover is really pretty common. There is normally one species that it is at competitive disadvantage. And Ian Rose, he had in mind forage crop mixtures. He was a breeder of white clover and concluded that compatible legume grass mixtures should have a high total yield and legume content large enough to optimize the benefits of its nitrogen fixation and superior forage quality. So already starting from the standpoint that normally the legume is at competitive disadvantage. Lisa Zannone, she used to work here in my same research center many years ago. She studied mainly intraspecific competition studies, and she concluded that the total yield of a binary mixture is maximized by pairs of components which in pure stand have similar yield and both of them high yield. Now, I verified these conclusions on the data of the large reference experiment I was talking about. And here you see the clover to grass pure stand yield ratio. So that when we have one, it means that their yield in a pure stand is similar. And indeed, we see a peak here of total mixture yield in the presence of values close to 1. And here you see the total yield of the mixture as a function of the average of clover and grass component yield in pure stand. So it's not anymore a matter of balance, it's just the average of the two. And indeed, there is a trend towards higher mixture yield when we have pair of components which individually have a high vigor, have a high yielding ability in pure stand, at least up to a point here. And a really crucial biological parameter when we talk about selection for intercropping is the genetic correlation between genotype performance in pure stand and in mixed stand. Unfortunately, genetic correlations are not frequently reported. Normally, people report phenotypic correlations, which suffer of the environmental effects, meaning especially that the experiment error could lead to, let's say, a bias in the assessment of the real relationships between responses, unlike the genetic correlation. So what I did here for this workshop is considering a set of studies that reported genetic correlations from pretty large number of legume genotypes. We have in blue green legumes, in red forage legumes, reporting the genetic correlation for genotype responses across conditions as a function of the mixed stand, pure stand yield ratio of the legume. Now, what I mean by that, reporting the legume yield in mixed stand to same unit area as pure stand, is that when we have 1, means that on average, the legume genotypes are producing in mixed stand as much as in pure stand, which is pretty rarely the case because normally legumes are at competitive disadvantage. And you can see here this trend, so that the greater the imbalance, the greater the competitive disadvantage of the legume, also the lower the genetic correlation. That is, the greater the need to select specifically under competition, to select in intercropping. You see with negligible difference between grain and forage legumes. And I also consider 2 studies in which there were phenotypic correlations, but since there were also broad sense readability values, it was possible to estimate the genetic correlation values. So that are indicated by this arrow. And here, for example, this is an alfalfa experiment, and under certain conditions, alfalfa is really far more competitive than grasses. And here is producing more than 2 times more in mixed stand. And you see that the estimated genetic correlation here is nearly 1. Finally, in a review paper of mine and other colleagues, and I'm also adding this recent paper, considering different studies, it was clear that specific mixing ability effects are usually much smaller than general mixing ability effects. Now, specific mixing ability, they are genotype by environment interaction effects when you consider the total yield in a factorial layout where we have grasses, for example, as columns and legumes as rows, no? So that whenever we have a given combination that is producing much more or much less than we would expect from their mean response across different mixtures, then this is a specific mixing ability effect. So in general, these combinations that are particularly successful or unsuccessful, they're really rare, unless we consider material with a history of coevolution. This was shown essentially for forages. So that there have been actually selection schemes that have been proposed when we are selecting specific pairs of components, meaning to have a couple of varieties that are marketed together. Well, Of course, it is questionable anyway, the practical interest of selecting and marketing pairs of cultivars that are indissolubly linked. No, because probably it is pairs of cultivars could have a pretty small market share. And for forages, for example, often we have mixtures with more than two components, so that this specific mixing ability effect could be diluted. And all farmers would like to buy different varieties from different seed companies. So that, let's say, actually probably there is more interest in marketing, in selecting these specific pairs of cultivars. And anyway, there isn't much ground to do that normally. So the overall evidence, I would say, is the key importance of selecting for greater competitive ability the species that is subjected to severe competition under the likely management, because greater competitive ability of the dominated species will raise the mixture yield. And for T species, the genetic correlation for responses of the genotypes across pure stand and mixed stand tends to be low. And as I anticipated, most legumes are ordinarily outcompeted. So clovers are from grasses, pea, lentils, welches from cool season cereals, soybean or common bean from warm season cereals, meaning that in many cases we need to select legumes for intercropping, specifically for intercropping. Now, one could think as an alternative, instead of breeding for competitive ability, to use a crop management that can alleviate the legume competitive disadvantage. And this is possible by adopting less vigorous genotypes of the companion species, reducing or avoiding the nitrogen fertilization, increasing the legume sowing density, or using spatial arrangements that reduce the competition, like strip sowing. But most of these solutions would decrease the mixture yield or reduce, like here, the potential benefit of intercropping. Now, the practical implications of the large reference experiment I talked about was, okay, first additional studies, but then this is the way we selected white clover here. So it was in a mixed stand using two vigorous varieties from different species. And this led to select the variety Giga. And indeed, the Giga represented a big progress for competitive ability with grasses compared to other material. You can see here Giga in the third year compared to a good reference variety like Espanso. You can see here the white clover has nearly disappeared, so that clover dry matter yield, average values across 4 grass varieties is much higher here. There is also a trend towards higher total yield, a clover proportion that is reasonable, no? 30% to have also high forage quality. And also there is a trend here towards lower weed proportion. And there has been also like a methodological comparison of selection in intercropping compared to, for example, selection in pure stand, phenotypic, followed by half-SIB-based genotypic selection in mixed stand, or directly phenotypic selection in pure stand. And although, for example, here we used a higher selection intensity, again, the yield response, the progress was much lower. Also here, where the selection was more complex because of the 2 stages. And we could use these studies with many genotypes also to assess the predicted efficiency of different selection strategies, so that comparing the direct selection for yield in mixed stand with indirect selection based on yield in pure stand, or also a selection index or useful traits in pure stand. These are the expected gains according to selection theory. This is the root square of the broad sense irritability in the target condition. This is the square root of broad sense irritability in the selection condition, and this is the genetic correlation between the two conditions. And in the end, we could express the results as relative predicted efficiency, that is, as a ratio of the two yield gains, equaling the former direct selection to 100 or to 1. And I'm reporting here the results for a forage legume and a grain legume. And you see that in both cases, pretty consistently, actually, we have a reduction of predicted efficiency when we select for yield in pure stand compared to direct selection. We lose about 50% of more of our efficiency. And because the genetic correlation is pretty low. And in addition, you see here the selection based on a selection index of traits in pure stand. Now, there were different traits, but one of the main ones was the canopy height, the plant height of the legume in order to better compete with light. And in this case, anyway, so selection in pure stand based on a selection index is more valuable than selection based on yield in both cases. It is particularly high valuable, I would say, nearly as good as direct selection when we consider P, because the genetic correlation here is higher, and also the irritability, you see in broad sense, for the selection index was high. So from this kind of experiments, we could have also an overview of what happens if we select depending on different scenarios. Now, getting a little bit more to the issue of the useful traits to select for. Indeed, often the important traits, the adaptive traits that contribute to yield in mixed stand are different to those for yield in pure stand. For example, here for P, for the production of grain yield, you see different traits that were evaluated in pure stand. And the grain yield in mixed stand of the P was correlated to later flowering of P only in mixed stand. And why? Because normally P is earlier than associated cereals. So this was important to have later genotypes. Or plant height, as I said, this trait is very important for competition for light. And here, the yield in mixed stand, but not in pure stand, is correlated. While the individual seed weight, for example, is important only in pure stand, and also the susceptibility to Ascochyta blight. This trait is far more important in pure stand than in mixed stand, where we have a dilution of the pathogen, so that really tolerance is more important here. And these results are also confirmed by this mixed stand, pure stand, P yield ratio. This variable refers to having relatively greater yield in mixed stand relative to pure stand, And you see that indeed relatively greater yield in mixed stand is associated to later flowering and taller plants. Now, when we consider the useful traits, we could have, I would say, two broad situations. First, we may have some traits that I would say we could reliably assess them in pure stand, because although they might be affected by plant plasticity at the species level, they are little affected by genotype by growing condition interaction. And then we have traits in which we have genotype variation for plant plasticity, so that we can have genotype by growing condition interaction. And I would like first to show you graphically two situations, two examples of these traits. Later on, I'm going to show you numbers. So here we have an example, P plant height, which belongs to the first type of character. There were 3 genotypes, for example, and genotype 3 is taller than the other 2. When we have intercropping, all of the genotypes have a taller stature to respond to the situation of competition, but genotype 3 remains the taller. While for white clover petiole length, that is for a stoloniferous species, it actually determines the canopy height. We have 3 genotypes. Genotype 3 in pure stand is taller, but when we get to intercropping, we have a genotype 2, which is actually taller, that is by plasticity at the genotype level can respond much better than the other to the situation of intercropping. And here you can see the numbers for a set of material, a pretty large number of genotypes, as you can see in both cases. So here you can see the mean value in pure stand and mixed stand of the 2 traits. In both cases, we have a taller canopy for white clover and P. But when we consider genotype by condition interaction for the trait, we have it for clover petiole length, and we don't have it for P stem height. The correlation of the trait in pure stand with yield in mixed stand is high anyway for both. And in addition, the correlation of the trait in pure stand with this mixed stand pure stand yield ratio, that is, this ability to go relatively better in mixed stand, is still significant and particularly important here. Now, we can consider a mixed stand pure stand trait ratio for these traits only when we have condition by genotype interaction for the trait, that is only here. And this partial plasticity trait of white clover, it is indeed further important for better stand in the competition by grasses. You see, this ability to make a relatively greater petiole elongation is then further associated with yield in mixed stand and with a relatively better yield in mixed stand of the clovers. Now, we could speculate why we can find variation, genetic variation for plant plasticity for this white clover trait, but not for the P height. Perhaps this is due to the fact that white clover and other forages, they have a long history of coexistence with the grasses, so that perhaps evolutionary, this kind of genetic variation had the possibility to develop. But the main point I would like to make is that genetic variation for plant plasticity, it could be useful, but it is difficult to exploit because it shows up only in mixed stand. That is a condition where we may already be assessing directly the yield response. So which are the useful traits to select for intercropping? I already mentioned several times plant stature or high relative growth rate, they are associated. So they contribute to higher competitive ability, especially under favorable growing conditions, for competition for light, according, one could say ecologically, to a scale foraging strategy of the genotype. Then we have plasticity traits like the petiole length trait I showed you, and They are useful more according to a precision foraging strategy, which ecologically means the ability of a genotype to make exploitation of in the canopy of areas that are particularly useful, where there is less competition for light, just by moving carefully in the canopy. For example, also stolon growth in white clover is another of such traits. Then the complementarity of growth pattern can be important, especially for biomass production in time. Then different plant architectures, different root distribution. But the point is that we know very little about these traits. So there is a lot of speculation, but we don't have still much evidence. For example, on the influence of different plant architectures, different root distributions, on the ability to better compete. For example, here in Lodi, we are studying root competition, considering white looping plants that are of contrasting type, not tall or semi-dwarf, etc. And also the indeterminate growth habit can be an important trait, which actually combines some features of the above traits, because it can contribute to taller plant stature, but also sometimes to plasticity, etc. You can find much more in this review paper that I wrote a few years ago. And at the end of this review paper, so we concluded stating that there is an urgent need to assess the efficiency of novel and relatively low-cost breeding strategies, among which evolutionary breeding and genomic selection emerge as the most promising. Now, I would like to show you our first results regarding genomic selection versus phenotypic selection. It is for pea grain in intercropping. And what you see here is a comparison in terms of actual yield gains in mixed end for pea yield, when there was 1 year used for selection for pea yield in mixed end, pure stand, according to a selection index, or to develop a genomic selection model, and then the other season used for validation, and this for each of the two seasons, so that in the end you could see the average of the relative efficiencies of the two seasons for the two selection criteria. And what we could see here is that direct selection in mixed end for pea grain yield is still has the highest efficiency. As I anticipated, selection in pure stand according to a selection index is anyway highly valuable, but the genomic selection as well has a high interest, because we have a little loss of efficiency, but in the end, the cost for the genotyping assessment of a genotype tends to be lower than the phenotypic assessment into replicated plots. So this is, I would say, indeed a promising selection strategy. And then regarding the evolutionary populations, here in Lodi we are developing a population of white looping NP in mixed end and in intercropping with cereals. And I would like to clarify that we could use these populations either to select then a pure line variety, so that we have actually an evolution under mixed stand across different generations, instead of having the typical single seed descent based selection. Or we could even use the evolutionary population as a real population into cropping. So we are keeping in our work both scenarios open. And today I am anticipating some data that we have just analyzed a few weeks ago. They are data averaged across 2 years, in which we have two populations from just the same genetic base, which evolved in mixed stand or in pure stand for 4 generations. And then when we assess the P yield in mixed stand and in pure stand, or the P proportion in mixed stand, for 2 years, well, what we found is a pretty striking evolutionary effect, I would say, because you can see that the population evolved in mixed stand showed definitely greater P yield in mixed stand and also P proportion, which is important, no? While the one that evolved in pure stand, sorry, here I realized that there is a mistake. This should be 90 and this should be 12. And while the one that evolved in pure stand, we have much lower yield here, but we have higher yield here in pure stand. So a real evolution towards specific adaptation to these conditions. And that was all. So I thank you for your attention, showing a picture of an intercropping in the balcony of my house. I would like to recall various projects that are funding our work on intercropping, and also our research group that is contributing to our work. Again, thank you.
Thank you, Paolo, for your contribution. And as always, after the presentation, there was room for question. If any of you want to directly ask, you can. Otherwise, you can use the chat. And okay, there's a question from Tom Workington. How do you manage weeds in intercropping?
Well, this is more, I would say, an agronomical problem, because there aren't many herbicides, let's say, useful for broad leaves, but are not killing, so at the same time, cereals or legumes. Meaning that probably intercropping should be seen as a agroecological cropping, in which we don't use herbicides. We normally don't use herbicides, but we just exploit the strength of the two crops to control the weeds. So first of all, it's very useful for organic agriculture. And anyway, as I showed, selecting for competitive ability for the legume also improve the ability of the legume and of the intercrop as a whole to control the weeds, to compete against weeds.
Okay. There's a second question from, I can only read Sebastian something, Sebastian Kazman. Okay.
Yes. Can you hear me?
Yes.
Yeah, perfect. I have 2 questions, and one is about the cropping partner. Actually, we have a breeding program for pea in mixture with barley, and we were actually, since a few years, we are wondering, because we are doing the selection with one genotype of barley, and we are selecting the peas from there, and we are actually wondering if it would be good to use also a mixture of barley varieties, because I'm especially wondering about the low below ground traits, so that because we have no idea what's actually happening with the roots, and if there's also some kind of niche specialization below ground. And then the other question we have is, like, last year we couldn't register a genotype, which is quite good in pure stand in mixture, because we cannot we do not pass the official trials in pure stand, and they are always in pure stand. And if you know about any examples in the official variety registration where a mixture, like official trials for the registration is possible in mixture. Thanks.
Yeah, Sebastian. So for the third question, I think you're right. I mean, although specific mixing ability effects are not expected to be large, I think one is on the safe side if it is he's using or she's using a mixture of different varieties as a tester, as indeed I showed for white clover, and also as we are doing for all the work on that I showed on grain crops. So, for example, the mixed stand was normally barley and wheat. When we had trials, evaluation trials, and the evolutionary populations are evolving with different varieties of different species. Of course, with a preference for material with a strong competitive ability. Now, your second question, I think I'm only aware of white clover in UK and probably in France, that it is evaluated in intercropping. But then perhaps in some registers of varieties, some national registers, it is possible to specify among the special conditions for evaluation or among the special traits that variety should be tested in intercropping because it is particularly useful for intercropping. This is possible in theory, for example, in Italy, although I've never presented yet a variety for intercropping. And in the future, I think we should foresee more and more, at least this possibility, no? That if one wants, this can be done.
Okay. Is the question, is the answer okay? Okay, so then we have Stefano Zanotto.
Yeah.
Hi, Stefano.
Hi, Paolo. Thank
Hi.
you for the interesting presentation and very comprehensive. I have a couple of questions. The first referred to your last slide about this evolutionary population. Maybe you said it, but I'm not sure. When you tested the pea, what was the other crop? It was grown together. What was the partner crop? And then the second question is more not really referred to your presentation, but I'm wondering if you have ever considered the idea of intercropping forage legume with a more traditional grain legume I mean, used normally as a grain legume to make forage mixtures, including different forages and grain legumes at the same time.
Thanks. So for the first, yeah, in that experiment, like in any other way, the associated cereal is also a mixture. Normally, it is a mixture of two varieties. Normally, we use one barley variety and one wheat variety for that. And so having different forage and grain legume material together. So I guess if one is considering a grain legume and a forage legume as a binary mixture, so actually I never considered this kind of intercrops. If we are considering binary mixtures, of course the situation is a little bit easier than when we have a complex mixture. No, when we beside the two forages, grain and grain legume and forage legume, we also have perhaps some other material like cereals, I don't know, oat or grass, etc. Of course, all that makes things more and more complex, no? But I would say it's always useful to consider the competition dynamics and to work on the species that it is at competitive disadvantage.
Okay. Stefano is okay with the answer. I have Dylan. I don't know how it's pronounced. Please.
We cannot hear. Dylan, would you try to switch on the microphone?
Yeah, you're muted, Dylan. Let me see if I can unmute. Nope. Dylan, we cannot. Okay. Now it's working.
Okay, sorry. I was just wondering, actually, because I'm intercropping peas and faba beans in Sweden, and I'm using a wide selection of pea varieties, but I use mainly old varieties and land races that is technically kind of forage peas, very vigorous, big. And I was wondering, in your lines of the pea lines and so on, if you include also this type of material, or if it's more like these semi-leafless types only.
Yeah, we are working on semi-leafless type because then anyway we should produce seed in pure stand, and a semi-leafless type would be more tolerant to lodging. And there was a slide that then I took away, not to make it too long, in which I was also mentioning the fact that we have studied a tall pea compared to a semi-dwarf pea, no? And for sure the tall pea was better able to compete with wheat, no? So that we have then registered indeed already as a variety this tall pea that is semi-leafless as a variety specifically for intercropping with cereals, especially for biomass production. And we were lucky that anyway it passed the value for cultivation and use test, although it was evaluated in pure stand. So yes, a tall material can be quite useful for intercropping, but tolerance to lodging is an issue that should be considered, because it is not an issue when we are cultivating, but it is an issue when we are producing the seed for sowing.
For me, it's quite interesting, because it's actually the intercropping for me is a way to cultivate this wide diversity of pea. So I use the cropping system more as a facilitator in this. But I also wanted to comment a bit on this whole thing with the VCU and the seed legislation and putting. I've been trying to communicate now with the new seed legislation coming in the European Union, the value of niche varieties and something that facilitates that we can actually put varieties for kind of alternative production systems. I don't know if anyone else have had the chance to communicate this in that process, but it feels like we're reaching a point where we see the value of alternative phenotypes that maybe wouldn't make it through in pure stands now. That was just a comment.
I don't know if anybody of the present can respond to Dylan about that.
Okay. I guess we leave Dinan's comment as it is, and we may move it. There's another question going on from Rina Dubey or Rena Dubey. Rena, you're muted.
First of all, thank you for the nice talk, Paolo. I have one question. Very interesting. I'm working on cereal and legume intercropping, but on triticale and fava bean. But my questions are, in one of the slides, you talked about the correlation, the phenotypic correlation between the grain legumes and the forage legumes. And also we know that the phenotypic correlation is very much affected with competitive stress. So what do you think? The correlation can be dynamic. It's not always the same range like we saw in the presentation. What are your views about it? Just wanted to know.
May I ask, Rina, as you mentioned the correlation, like for example, between yield or performance and then morphophysiological traits, right? Yeah, indeed, because the fact is that, okay, first of all, I gave an example, no, a clear example in which when we have a trait intrinsically assessed in pure stand, so without the effect of competition, then already its relationship with performance in pure stand and mixed stand can be very, very different. No, this is already something. Then, of course, when you go to consider the same morphophysiological traits in the condition of mixed stand, the presence of genetic variation for plasticity, if there is, could further confuse the situation, no? And but I think just to keep it simple, I would rather consider so unless you are thinking to exploit the genetic variation for plasticity, which is anyway useful really to study just theoretically.
Indeed.
But then what really matters, I think, is the assessment of the trait itself in pure stand, intrinsically, let's say, with the performance in mixed stand. But yeah, as you said, it
can be dynamic. Yeah.
Absolutely.
Yeah. Indeed. Yeah, and one more question. What are your insights about the multi-location intercropping trials? Do you have any insights for them? Because most of the intercropping experiments are at single location, and then we know, okay, this is the interaction, this is the competition, and those are often with the limited number of variety combinations. So any insights from there? What could be the effect if we do multi-location intercropping trials?
If we do, it would be much better, so that we could also see the interaction with the location. All I managed to do in my life, I think, is considering different years, which is anyway very, very important.
Indeed.
No? When we have annual crops, but would be better. The point is, when you have like an agronomic trial with a pretty low number of combinations, you can easily have a multi-locational trial. But our view, no, was always much more as breeders. And then we need large numbers, and then it really gets complex if you have, besides different years, also different locations.
Indeed. Yeah. Okay. Thank you.
We have a comment from the chat, which is more, I guess, with a reference maybe with to Dylan's comment before from somebody that I cannot really pronounce the name, from Kien Nguyen from Vietnam National Plant Gene Bank, saying that they are interested in how to measure and track benefits of intercropping between legume and other crops to increase resilience and low emission. There's also a link to their ResearchGate webpage, seems like. So check the check the chat if you are interested. Are there any other questions or raised hands? Doesn't seem so. So again, I thank everybody for being here. And as usual, the recording will go up online in a few hours or tomorrow at most. Okay, thank you all, and see you all in a month for the last webinar for this season that will be in early June with Jose Carlos Jimenez. Bye.
Thank you. Bye.
Goodbye, everybody.
Thank you. Bye-bye.